This is Chapter 6, Section 5, and it's on traits and probability. And the key concept here is that the inheritance of traits follows the rules of probability. All right, a Punnett square illustrates genetic crosses. So a Punnett square can be defined as a grid system for predicting all the possible genotypes of offspring. So, this wordy definition of a Punnett square isn't really as useful as the picture definition, the picture concept where we take two alleles, and remember alleles were just different versions of a single gene. And it has to be the same gene. It's a gene that's on homologous chromosomes, where meaning chromosomes that have the same genes. And so an allele would be this capital A right here and this lowercase a would be different alleles. And these alleles code for different phenotypes, where this genotype right here will represent a phenotype or some physical trait. And then the boxes in this Punnett square are going to show the genotypes of the offspring. So these are the parents, so parent 1 and then parent 2, and then these are the genotypes that result. And the Punnett square is going to yield the ratio of the possible genotypes and phenotypes. The idea of a ratio is something I'll explain what that means just by going through different Punnett squares, and you'll kind of get an idea of what I mean by that. All right, a monohybrid cross involves one trait. So a monohybrid is whenever you're using a Punnett square to cross two parents. One plant and another plant, and you're only examining one specific trait. So here we're examining the trait of flower color, where we have a homozygous dominant plant. Remember, homo meaning same, and dominant referring to the trait. The dominant's referring to the allele where you only need one of those alleles in order for that phenotype to be present. So we have capital F, and then the recessive parent right here, homozygous recessive, and recessive is where you need both copies of the alleles in order for the phenotype, in this case the white flower, to be present. So whenever you cross these together, what you do is these two alleles that kind of border around this square right here are going to meet up inside the box. So big F, little f, little f. Then you're going to go across where you take big F and little f and you put it in here. Big F, little f. Same idea here. Big F, little f, little f. Here, big F, little f, little f. So you get the result of this cross here, this monohybrid cross, is you're going to have heterozygous, and the hetero is coming from where you have two different kinds of alleles, the big F, the dominant, and the little f, the recessive, where you have the dominant purple color that results. So all of them, so the homozygous parent, the homozygous dominant parent, the homozygous recessive parent, whenever they cross, you create all heterozygous, all big F, little f, and all dominant phenotypes. Okay, so then there's another, if you take then the heterozygous parents, different combinations of parents, you're going to get different results. So you have big F, little f, which is still purple, and big F, little f, which is still purple. And you take big F, you get that. And you have big F, little f, and you get a heterozygous, but it's still dominant for the phenotype. And big F, little f, you have another heterozygote, but it's still dominant for the purple phenotype. And then you have little f, where this one's going to be homozygous recessive, where you're going to have the recessive white color. So you took the heterozygous parents, and you created a 1:2:1 ratio of the genotypes, meaning the 1 is referring to having one big F for every two big F, or heterozygotes for every one little f, homozygous recessive. And then the phenotype here is going to be 3-to-1, dominant to recessive, because you have 3 of the dominant phenotypes for every 1 of the recessive phenotypes. Okay, and then if you take the final different monohybrid cross that we can do, you have up here, you have the homozygous recessive, and then the other parent's going to be the heterozygous. And what you're going to get is you're going to get half of them are going to be the heterozygous purple flowers, and then the other half's going to be the homozygous recessive white flowers. And so that's going to look like 1-to-1 for the genotype, where you have for every 1 heterozygote genotype, you have 1 homozygous recessive genotype, and then you're going to have 1-to-1 for the dominant recessive phenotype, because you, for every 1 purple flowered dominant, you have 1 white flowered recessive. The idea of a test cross. So a test cross is whenever is a cross between an organism with an unknown genotype and an organism with a recessive phenotype. Meaning that this homozygous recessive parent can kind of, imagine that if I crossed out this, where we didn't know that this was, we didn't know what this parent was, and this was like X or something. This homozygous recessive, if you know what the offspring are, you can work backwards, almost like an algebra problem, to find out what X is. So if you get your offspring here where you have, heterozygote, where it's this 1-to-1 ratio, then you must know that this X here has to be big F, little f, because that's the only thing that's going to produce this monohybrid cross. Conversely, if everything here was big F, little f, if all the offspring are big F, little f, and they were, all purple flowered, then you know that the X would have to be big F, big F. Or if all the flowers were white, then you'd have to know that, oh, this parent has to be little f, because that's the only thing that would allow all the flowers to be white. So test cross is whenever if you want to find the identity of the unknown parent, you can do that using the homozygous recessive parent, assuming you know the identity of all of the offspring. Okay, so don't freak out. This is much easier than it looks. When you do a dihybrid cross, that's when you have 2 traits, hence the prefix di right here. So Mendel's dihybrid crosses with the heterozygous plants are going to yield a 9-to-3 to 3-to-1 phenotypic ratio. Now let me explain what that means. So imagine, the dihybrid cross, you're doing 2 traits. Here, they did the trait for color, which they used Y for, and then shape, or the P, which they used as the R. So we crossed 2 heterozygous parents where they were heterozygous for both traits. So they were big Y, little y for color, so yellow, because yellow is a dominant here, and green, little y is recessive. And they crossed that, and then the other one was big R, little r, which is heterozygous for the round shape, where little r is going to be the wrinkled. And same thing over here. So to do this, you have to make a Punnett square that's 4x4, so that you have 16 boxes in here. And then to set it up with the parents up top and on the sides, you're going to pull the first letter, so this Y, and this first letter, which is R, and you put that right here. Then you're going to pull the first letter and the second letter and do little r. Then so basically you're gonna create all the different combinations possible of these 4 letters. And same thing over here for this parent one. And then to cross them, it's very similar to what you do with the monohybrid cross. You take the Y and the Y, and you're going to put that first, and the R and the R, and you're going to put that second. Then you jump over here, Y, Y, that, maybe do this bottom one down here, little y, then that, and I won't go through all of them, but, as you fill it in, you'll have all of your different genotypes. And I'll point you back here to this 9 to 3 to 3 to 1 phenotypic. Remember, phenotypic is the physical features ratio, where you have 9 of your yellow and round phenotype and 3 of your yellow and wrinkled, and then 3 of your green and round, and then only 1 of your green and wrinkled. And what this ratio tells you, and what that allowed Mendel to conclude, is that and what it led to is his second law, which was the law of independent assortment, which is something we talked about when we talked about meiosis in the previous chapter. And so the law of independent assortment states that allele pairs separate independently of each other during meiosis. So to remind you of the first stage of meiosis, because remember there's a meiosis 1 and then a meiosis 2 where you're going to be separating the chromatids. But in meiosis 1 is when you have the law of independent assortment. And it's referring to this part right here in metaphase 1. So imagine that this right here, these 2 chromosomes were like our color, the seed color. And then these 2 chromosomes right here were referring to the seed shape. What the law of independent assortment says is that how these chromosomes right here, the ones that determine the color of the seed, how they travel in meiosis to the 2 different cells, is independent, meaning not dependent upon how the chromosomes that determine the color, these 2, travel. So, if this green chromosome goes into one cell, it has no bearing. No bearing on whether this red or this blue chromosome goes somewhere. Same thing with the purple one. They go their separate ways regardless of how somebody else goes there, regardless of how the other chromosome goes its own ways. Okay, so final slide, and that's going to be heredity patterns can be calculated with probability. So there's a lot of words on this slide, and it's basically reviewing just the idea of probability. And probability is just the likelihood that something will happen. What is the chance you think something's going to happen? And probability is going to predict the average number of occurrences and not the exact number of occurrences. And what that means is, imagine you flip a coin 10 times. On average, what probability would say is that you should get half of those 10 tosses should be heads, so 5, and the other 5 tosses should be tails. However, what usually happens, if you were to do it right now, you might get 8 heads and only 2 tails. But what's going to happen on average, so if you did it 1 billion times, you're eventually going to get to a point where you have half of the time being head and half of the time being tails. And this is the formula for probability. So it's the number of ways a specific event can occur. So with flipping, for flipping a coin would be 1. And then the number of total possible outcomes, which with flipping a coin it can either be heads or tails. So you have 1 over 2, which is 50%. Okay, and so, why do we care about that is because probability applies to random events, which is what meiosis and fertilization are. So whenever you're doing meiosis, whenever you're creating the sperm and the eggs, that's a random event. You don't know what the genetic makeup is going to be for each individual sperm or each individual egg, and then you don't know which sperm is going to fertilize which egg. So those are all random processes. Okay, and that's going to do it for this section.